We're now going to talk about glucose and carbohydrate metabolism in general. I have about a few minutes to sort of go over the overview, so I probably won't get to everything. This over here on the left, extracellular matrix and cell wall polysaccharides. We don't have cell walls in human cells, but this is relevant for plants. So don't get confused. If we're talking about this kind of part of the slide is really only relevant for plants. But what we're going to focus more so on is humans, because that's, I think, what you're interested in as future health professionals. So glucose can go to a few different possible pathways. Can form glycogen in the liver or in the muscle, are two places where glycogen is stored. And as we saw from the G protein beta adrenergic receptor reaction in the previous slides, the breakdown of glycogen is really important in fight or flight response. So there's two places where glycogen will be stored. One is in the liver and one is in the skeletal muscle. Glucose can also go through glycolysis. Gosh, you've known about that since beginning biology, I'm sure, and maybe even from high school, or I hope from high school. So glycolysis is one other place glucose can go. And then another pathway that students, I often call the orphan pathway, because unfortunately students always forget about it, is the pentose phosphate pathway, which serves to produce a precursor for those really important nucleotides, DNA, RNA, etc. So here's glycolysis. You notice that in the beginning we have some investment of energy. In the end, we end up producing some energy. The net, of course, is 2 ATP overall, but it does require some investment here. I'm going to look at, let's actually go back. Yeah, okay, sorry. I'm going to start with this one. So here, this first reaction definitely requires energy. It's catalyzed by hexokinase. One of the major regulation steps is this one here, which is catalyzed by PFK1 or phosphofructokinase 1, the enzyme named, by the way. So that's a huge regulation step. Another regulation step is pyruvate kinase, which is that last step in glycolysis. Often I hear students say that glycolysis sucks, and actually thermodynamically it kind of does, because the pyruvate kinase reaction at the end here is highly exergonic, and so it's really causing, through Le Chatelier's principle, all of the arrows to shift downward toward the product of glycolysis. So if you think glycolysis sucks, I'd say thermodynamically it certainly does, because of the thermodynamics of the pyruvate kinase reaction, which is highly ionic, and therefore shifts all the arrows through Le Chatelier's principle down toward the production of pyruvate. This is showing on the right the gluconeogenesis pathway, which is not an exact reversal of glycolysis, because there are a few in blue bypass enzymes that are necessary in order for us to go in the reverse direction. In other words, hexokinase, PFK1, and pyruvate kinase, which not coincidentally are the thermodynamically exergonic reactions, they're not reversible. So we have to have bypass enzymes in order to go in the reverse direction. And so those are labeled in blue. Importantly, you should remember that gluconeogenesis only takes place in the liver. Do not ever tell me or anyone else that gluconeogenesis will be active in any tissue other than the liver. Importantly also, when we're thinking of the break of glycogen and it being released into the bloodstream as glucose, that only happens in the liver, and that's as a result of glucose 6-phosphatase being available. The skeletal muscles that have glycogen and break it down will break it down into glucose 6-phosphate, which can then just funnel directly into the skeletal muscle glycolysis pathway. Whereas in the liver, you're releasing glucose. You can't release G6P, right, because of the phosphate group. But in the liver, you're actually releasing glucose into the bloodstream through this pathway. And so that's why you have to have this additional enzyme, this glucose 6-phosphatase in the liver, which is not present in muscle. Okay, here's that wonderful pentose phosphate pathway, which I just have to mention is a wonderful source of NADPH and also a precursor for ribose 5-phosphate, which goes on to make nucleotides, DNA, RNA, etc. Here's our glycogen synthesis. I skipped this, which is more about the pentose phosphate pathway, but in the interest of time, here's glycogen. We talked about, excuse me, in that G protein slide and the cascade that followed, the breakdown of glycogen into G1P, and this is how it happens, through glycogen phosphorylase and then a debranching enzyme. And that's because, remember, glycogen is highly branched, and the reason for that is because of the surface area to volume ratio is maximized that way. If we have a highly branched molecule, it's easier to store than to having like one really long molecule or a few really long molecules. So you get more bang for your buck, so to speak, that way. Another way to say that is the surface area to volume ratio is maximized. That would be a more scientific way of saying the same thing. All right. Okay. Phosphoglucomutase is the enzyme that converts G1P, which is the product of glycogen breakdown, into G6P, which is necessary because G1P, there's nowhere for it to go in terms of glycolytic pathway. We have to convert it to G6P in order to funnel it into the glycolytic pathway. I mentioned in the liver, there's an enzyme called glucose 6-phosphatase that results in G6P being converted to glucose, which then glucose can be sent out into the bloodstream. Again, the liver and the muscle have very differing functions in terms of how glycogen is utilized and why it's there. The liver is basically storing glycogen as a source of glucose so that it can monitor the blood glucose concentrations and send glucose out as the blood needs it, because it's really important that the bloodstream have a certain concentration of glucose. If you get below a certain concentration, it will have dire effects on the human. Certainly after you get past a really low concentration, you can end up in a coma and you can die from having a low glucose concentration. So that's why the liver storage of glycogen is so important. In the muscle, it's more prevalent or more important for the fight or flight response, which we kind of already talked about. So we then, on the other hand, have to be able to synthesize glycogen, right, in the liver and also in the muscles. How is that done? So just showing that there has to be a way of synthesizing it. There's an enzyme called glycogen synthase. We actually have to activate a glucose with a UDP. I didn't go into all that detail, but UDP glucose will then be added to a molecule of glycogen through the actions of glycogen synthase. We also then not only have to make the linear part of the molecule, which have alpha 1,4 glycosidic bonds, we also have to make the branch points, which have alpha 1,6 glycosidic bonds. And that's done through glycogen branching enzyme, which is very easy to remember, unlike some of the other names of some enzymes. Okay, this is a nice summary for anyone who is interested in knowing under high blood glucose how this actually works. Here it actually shows insulin activating different proteins, which results in an increase in glycogen synthesis under high glucose concentrations in the liver, right, and also in the muscle. And low blood glucose concentrations, glucagon is the hormone at play that's active. And then as a result, at low blood glucose concentrations, we're going to have more glycogen breakdown in the liver, not any synthesis. You're never going to have synthesis and breakdown happening at the same time in the same tissue. That would be a futile cycle. So always these regulations are at play or have been put in place to prevent that from happening. So this is the overview, right? We've got glycolysis. Next, we have the pyruvate dehydrogenase complex that creates acetyl-CoA. Notice the pyruvate dehydrogenase complex is in the mitochondria. So pyruvate actually transports into the mitochondria to be acted upon by the pyruvate dehydrogenase complex. This reaction, the pyruvate dehydrogenase complex reaction, is highly exergonic, cannot be reversed. So if you have acetyl-CoA, it cannot go back through gluconeogenesis in the liver. It cannot. Pyruvate can, but acetyl-CoA cannot. Oxaloacetate is then condensed with acetyl-CoA to make citrate. We go through the citric acid cycle. What's the point of that? We make 3 NADHs and 1 FADH, and then both of those can go through the electron transport chain directly. We also make, I think, 1 GT, depending on which textbook you look at, it's either 1 GTP or 1 ATP in one of the steps. But the major source of potential energy is this NADH and FADH2, which can go directly into the electron transport chain. So I'm gonna skip over some of the details, but I've put them there in the slides for you to look at, different regulation points, some of the math of how many ATPs are produced. And I'm gonna briefly just look at this electron transport chain slide, and then we'll end the PowerPoint, we'll go to the problem set, and then we'll end with some cars, okay? Again, I don't think I'll be able to get through every single problem, so I'll start at the top of the problem set, and then probably in the end, you'll look at the solutions and then check them out, and hopefully they should be pretty clear. I tried to make the solution set pretty clear. All right, so we have an inlet point for NADH in complex 1 within the electron transport chain. Complex 2 actually is succinate dehydrogenase of the citric acid cycle. So this is actually a citric acid cycle reaction. This complex 2 is actually an enzyme as well. It converts succinate to fumarate within the TCA cycle. That's where your FADH2 is coming in, is in complex 2. NADH is being put into complex 1, and as a result, what is really happening within the electron transport chain is we're moving electrons from one discrete location to another, to another, to another, to another, and those discrete locations have different standard reduction potentials. Basically, what we're doing is we're moving the electrons from one standard reduction potential to another, to another, to another, and in so doing, each time we're releasing a small bit of energy. Each time it goes to a different standard reduction potential, it's releasing a small bit of energy. And that energy is then going to be harnessed and coupled, really, with the pumping of protons over across this intermembrane space, or sorry, into the intermembrane space is a better way of putting it, but across these complexes into the intermembrane space against the gradient, right? That's why it takes energy. We're pumping it against its gradient. And that is all fueled by the energy that results in the electrons coming in and moving from one to one. It's usually an iron sulfur cluster or a heme cluster, but moving from one of those discrete locations to another, to another, to another, to another. They're not depicted. That's what's happening, is we're releasing bits of energy and then harnessing it to move the hydrogen ions across against their gradient. Now, the number of hydrogen ions that are pumped, you'll see sort of slightly different numbers within each textbook, so be a little careful. I would say go with the numbers that are in the most common books. So like Kaplan would be a good resource. I don't know if those are the numbers that I'm using here, so just double check before you commit to it. But different textbooks have different numbers, and there are reasons for it, and we won't get into that. But in any case, just be careful with that. So the pumping of the hydrogen ions across against their gradient is occurring, and as a result, we end up with a chemical potential across the membrane. We end up also with a change in pH, because you know that when you have more of a positive charge, it's going to be more acidic. And we have more of a negative charge. We have an electric potential as a result of the chemical potential, which is the change in pH, the difference in pH across the membrane. We also have an electrical potential, which has more to do with the difference in charge across the membrane. As a result, there's this super cool protein, which you may have learned about, that has like this cool rotational mechanism, which I'm not going to go into huge detail, but as the ion is moved across and goes through that really cool protein, it causes it to rotate just a little bit. And every time each hydrogen comes in, it rotates a little bit more, kind of like a little record player almost, if you ever even know what that is. I mean, I didn't have record players when I was a kid either. It was before me. But anyway, you probably have seen them in movies and things. Every hydrogen ion that comes in, we have more and more rotation of the F0, F1 ATP ACE. Which is cool protein here. And with each rotation, we end up with sort of a change in conformation. I'm really summarizing here, but that results in the ADP within the matrix of the mitochondria being phosphorylated to ATP, and ta-da, we have our little exciting finale of making energy. So that is kind of the really fast overview of the key points, I think, of metabolism. I hope that you can spend some time with the slides, and also, of course, correlate it with the books that you're using, whether it's Kaplan, whether it's Princeton Review. I don't know if now the MP program has kind of their own thing, their own publication or whatever, but whatever study materials you're using, make sure that you sort of correlate when you're looking at the slides, correlate it with what the study materials are. Like I said, there are a few different sort of things like the hydrogen ions, numbers might be a little different. I think this is the most common numbers used, but nevertheless, check out the study materials.